All right, so today we will be talking about the two-hybrid assay method. This method is often called the yeast 2 hybrid screening, as it is often used in yeast cells. The technique is used to detect protein-protein interactions, and the transcription of a reporter gene or lack thereof detects whether the proteins in question interact or not. The two-hybrid assay method uses two proteins of interest, the bait protein and the prey protein. If they interact, there is transcription of the reporter gene. If they do not interact, there is no transcription of the reporter gene. The two-hybrid assay utilizes the biological system of transcriptional activation by engineering a transcription factor that binds to a promoter region, recruits RNA polymerase II, and transcribes a reporter gene. The engineered transcription factor is made up of two parts, hence the name two-hybrid assay. The DNA binding domain, which binds to the promoter region, and the activator domain, which recruits RNA polymerase and in turn transcribes the reporter gene. These components are each linked to the proteins of interest, your bait and prey protein. There are two possible outcomes for this assay. The first being that the DNA binding domain attaches to the promoter region. The protein linked to the activator domain interacts with the protein linked to the DNA binding domain. This would then recruit RNA polymerase II, which would then transcribe the reporter gene. The second outcome would be if the DNA binding domain attaches to the promoter region, the protein linked to the activator domain does not interact with the protein attached to the DNA binding domain. Therefore, there will be no recruitment of RNA polymerase and no transcription of the reporter gene. When actually creating this experiment, first, a reporter gene that can easily be observed or selected for must be used. For example, the HIS3 gene, which is required for histidine biosynthesis. Second, the plasmids containing your bait protein complex and your prey protein complex must be engineered. The bait plasmid will contain the code for the DNA binding domain, the bait protein, and a gene for selection, often a gene for the biosynthesis of an essential amino acid. The prey plasmid will contain the code for the activator domain, the prey protein, and another gene for selection. Both plasmids will be transformed into yeast cells in growth medium lacking the essential amino acids used for selection. Therefore, only the cells containing both plasmids will grow. Finally, the cells will be placed on growth medium lacking histidine. If there is culture growth, we can conclude that there was interaction between our proteins of interest, and if there's no growth, we can conclude that there was no interaction between our proteins of interest.